The development of approved vaccines for COVID-19 has been unprecedented, both in terms of speed and the number of different successful vaccines being approved. How has this happened so quickly? Normally, pharmaceutical companies will wait until a drug is almost through testing before they begin scaling up for manufacturing that drug. Because of the urgency and investment involved, the vaccines are being manufactured while still in testing so that they are ready as soon as they are shown to be safe and effective. This way of doing things is financially very risky, but saves many months. The speed to market is also thanks to all the work of previous scientific efforts, which gave researchers a safe foundation to build from. COVID-19 is only the latest in a series of novel coronaviruses that have emerged in recent years. The name comes from the crown-like shape of the protein spike that attaches to host cells to spread the virus. This protein spike has been the subject of research for years, following the outbreaks of SARS in 2003 and MERS in 2012, and this is what has given researchers around the world a head start in being able to produce vaccines. So how does the COVID-19 virus work? It is remarkably similar to other coronaviruses, including the common cold. All coronaviruses are covered in spike proteins, and it is these that are used to enter human cells. Inside the virus is the RNA, which is like a recipe for the creation of new viruses. A distinct portion of this RNA is responsible for reproducing the spike proteins. When a virus lands on a human cell, the spike proteins allow the RNA in the virus to enter the human cell. Once inside the cell, the RNA or messenger RNA begins replicating the virus. Each section of the mRNA is a recipe for a part of the virus. The human cell then dies, releasing the new viruses to begin the cycle again. So how does our immune system work? Well, it is the spike proteins that are the key. As soon as our immune system meets a new virus, it identifies the new spike proteins and begins to build the manufacturing process for new antibodies. As soon as these antibodies are produced, they begin to attach to each spike protein. This means the virus cannot enter a human cell, so it eventually dies without replicating. It is the time taken by the immune system between finding the new virus and manufacturing new antibodies that is the difference between getting sick and not. So how do messenger RNA vaccines work? Each part of the RNA is responsible for reproducing a part of the virus. This type of vaccine removes just the part of the RNA responsible for the spike proteins and wraps it in a protective, oily casing. It is then injected into us and the RNA enters a human cell. Once inside, the messenger RNA reproduces the only thing it can, spike proteins. When the cell dies naturally, the spike proteins are found by the immune system and it begins to build the manufacturing process for new antibodies. So, if and when a real COVID-19 virus is found, our immune system can immediately produce loads of antibodies and kill it before it reproduces. So, how do adenovirus-based vaccines work? This type of vaccine takes the part of the virus RNA that is responsible for reproducing the spike proteins. Then it makes it more stable by attaching it to some DNA from a sample virus that doesn't affect humans. The adenovirus is then injected into us and it is engulfed by the human cell. It then makes its way to the cell nucleus and the DNA enters it. Once inside, the DNA produces mRNA, which reproduces the only thing it can, spike proteins. When the cell dies naturally, the spike proteins are found by the immune system and it begins to build the manufacturing process for new antibodies. So, if and when a real COVID-19 virus is found, our immune system can immediately produce loads of antibodies and kill it before it reproduces.